Hi guys, Dr. Orlando here, going to do a video on glucose breakdown and ATP production. This is what we call the big picture in class, the one that took up the whole board that we drew. So go ahead and get that out and be ready to look at your notes as I go through this video. Okay, so we're going to talk about harvesting ATP from glucose. So we know glucose is C6H12O6, and we're going to draw glucose linearly. So you can see there's those 6 carbons all in a row. Remember, these carbons are covalently bonded, and that the energy is stored in those covalent bonds. So that's our potential energy in glucose. We're going to break it down and turn it into kinetic energy in the form of ATP. Okay. So the first thing that's going to happen to glucose is it is going to go through glycolysis, and that occurs in the cytoplasm of the cell. First thing is glycolysis. Okay, and from glycolysis, we will have a breakdown product. We'll look at this on the next slide here. We'll have breakdown products, and that product will either go through fermentation, which would occur in the cytoplasm, or it can go into the mitochondria where it can be further broken down to get more ATP. If it goes into the mitochondria, it's going to first have to go through a step we're going to call transition. And in transition, it just gets ready to enter the Krebs cycle, 7-step cycle. And then after that you're done with the carbons in glucose, but there's another step to harvest energy, and that step is called oxidative phosphorylation. And so that's made up of ETC, which is the electron transport chain, and chemiosmosis. Okay, all right. So we'll go on to the next slide here. All right, so the first thing we're going to talk about in a little more detail is glycolysis. Glycolysis, as I mentioned, happens in the cytoplasm. It does not require oxygen. So if the cell is in an oxygen-deprived state, it will use glycolysis to produce ATP. If it is a low oxygen environment, the oxygen may still be used to produce ATP in the mitochondria, but they'll have a backup of product in the cytoplasm, so some of that will might go into fermentation afterwards. Okay, but the process of glycolysis is the starting step here for either going into the mitochondria or going into fermentation, and it does not require oxygen. It is a low ATP yield, so we've got this 6-carbon glucose, and we'll see we're only going to net, get a good color here, we're only going to net 2 ATP per glucose. So that's a pretty low yield. Okay, so let's move up here and have a look at this process. So there's our glucose again, C6H12O6, and you can see drawn it linearly here through a process called substrate level phosphorylation, and we should review our notes on this as well. With substrate level phosphorylation, you take a phosphate from a high energy donor and you stick it onto ADP and you get ATP. Okay, so that's what happens in glycolysis. Through a multi-step process, we take phosphates, we add them onto substrates, and then later we harvest them. And we have to put 2 ATP in, and we end up getting 4 ATP out. So that is where we get that net 2 ATP. Okay. What else do we get from glycolysis here in the cytoplasm? Well, remember these guys here? Let me zoom in. NAD+. This is an electron carrier. Remember, I always draw him as a little sad guy because his backpack is empty, right? He has the positive charge there. So we would say that this guy is in an oxidized state. Let's write that in there. Okay. during glycolysis, we take those electron carriers and we reduce them. So this guy here, he's happy because he's got his electrons now, he is reduced or has gone through the process of reduction. So he is reduced. And that's pretty easy to remember that he's reduced. He picked up electrons. I know, and I always draw them as one little electron there. But electrons are buddies, and they always run in pairs. They just don't fit. The two electrons don't. The letter E's don't fit in the backpack that I draw. But we know there's two of them in there. And he does pick up a hydrogen as well. You can see right there. Okay, so there he is carrying electrons, and we say again that he is reduced because it is more negative and has picked up an electron. Okay. So from glycolysis, after we have harvested the ATP and we have reduced our electron carriers, so now they're carrying electrons, we end up with 2 molecules of pyruvate. 2 molecules of pyruvate. So these guys right here. And pyruvate is a 3-carbon molecule. So you got 2 of them. Basically, you just snipped glucose in half through a multi-step process. All right, and that is the end of glycolysis. From there, the pyruvate has a choice. If there's enough oxygen present, it will be shipped to the mitochondria. To the mito. Okay. If there isn't enough oxygen present, then pyruvate will have to be converted to something else so that we can oxidize our electron carriers. So here you've got the 2-pyruvate. We're going to follow fermentation here, and then later on we'll do what happens if we go to the mito. So here's our pyruvate, and we'll say that we are in a no oxygen state or low oxygen state, so pyruvate's starting to back up. So we got to do something with it. So it goes through the process of fermentation. Okay. The purpose of fermentation is to unload those backpacks, to reduce pyruvate to lactate so that NADH can be oxidized. So it's a oxidation reduction reaction. So let's see what happens here. So here are these 2 guys. We filled up their backpacks over here, right? And now we're going to take them and we're going to unload the backpacks, which means we're going to oxidize them. So the electrons and hydrogens are removed, so you end up with NAD+. Okay, so now they're oxidized. And lactate, although it's still a 3-carbon molecule, is actually reduced, so it has picked up those electrons. Okay, so those are lactate, and then the lactate will be dealt with by the cell. Sometimes this can accumulate and cause pain, like lactic acid buildup in muscles. Okay. So why did we need to unload those backpacks? Well, because these guys have a job. They're constantly carrying electrons. It's their job. They're electron carriers. So what's he going to do? As soon as his backpack is unloaded, he's going to go back up here to glycolysis, to the enzymes associated with glycolysis, and start assisting. So as hydrogens are removed, this guy will pick them up. Okay. And so as electrons are removed, he will pick them up. So that's the reason that we have to oxidize NADH, so that we can remove those electrons off of the carrier, and he's able to go pick up new electrons and to keep this process going. Okay? No more ATP is gained through fermentation, only regeneration of electron carriers. So that's fermentation. So now let's go on to what happens if we have oxygen present. So we're going to go over here to the mitochondria, and we see a whole bunch of stuff there. We're going to zoom in. Well, you can see the big process before I zoom in. Remember, we're going to talk about what happens in the mitochondria, so transition, Krebs cycle, electron transport chain, and chemiosmosis is going to occur down here. Okay? All right, so we'll do this step by step. I know there's a lot on this drawing, but I'm going to zoom in on each step so that we can talk about it. All right, so here comes the pyruvate. Remember, pyruvate is a oops, undo that. Get back to having, is that right? Well, there you go. Transition. A pyruvate is a 3-carbon molecule. And the first step in the mitochondria we're going to call transition. Okay, so let's zoom in on transition step. Okay, so just looking at that. So what happens is pyruvate joins up with enzyme CoA. I drew him as blue, so you remember he's an enzyme. It's a big, giant molecule. Remember I said it was like a ticket. It's a ticket that pyruvate needs to get into the Krebs cycle. Okay, and the Krebs cycle is like a ride, right? So to do that, pyruvate is actually going to lose one of its carbons, and that will become carbon dioxide. And when we remove carbons, we are able to also harvest some electrons and hydrogens. We broke a covalent bond. Things are coming out. Electrons and hydrogens coming out. So who's going to pick up those electrons and hydrogens? Of course, our electron carriers will. So NAD plus will pick up the electrons, the pair of electrons plus a hydrogen, and he will become NADH. And now he's a reduced electron carrier, and we have a 2-carbon acetyl group. I didn't label this guy here. Hooked to CoA. And so let's put his name there. We call that acetyl-CoA. Acetyl-CoA's job is to, we zoom this way, we can see that acetyl-CoA is going into the Krebs cycle. Okay, that's what my big letter K is over here. Okay, so let's follow acetyl-CoA. Okay, acetyl-CoA being comprised of the 2 carbons that were left from the pyruvate, and a CoA, which I said was like the ticket to get onto the ride, and the ride is the Krebs cycle. So acetyl-CoA will join the Krebs cycle here between step 7 and 1. Step 7 on the Krebs cycle, and it's kind of strange to start with step 7, but it's a cycle, so you can pretty much start anywhere when you talk about it, but this is a nice place to start. So in step 7, oxaloacetate is formed. It's this guy here, and he's a 3-carbon. Oop, not 3-carbon. He's 4-carbon. I'm missing one on him. Okay, there, I fixed him. He's a 4-carbon intermediate. And so in this first step, acetyl-CoA joins with oxaloacetate to make citrate. Okay, makes citrate right here in step 1. Notice that citrate does not have CoA in it. CoA was just the ticket. Brought it to the Krebs cycle, got it, was able to be used to join oxaloacetate and citrate. CoA will be recycled. Okay, so he's going to go back and be recycled and be a ticket for another pyruvate. Okay. All right, so now we're into the Krebs cycle here. Let's put this more center. We're going around the Krebs cycle, and we're back up to a 6-carbon molecule, and through a multi-step process, and we looked at these steps in detail, remember, in class, and we talked about important enzymes, so make sure you cover those. But generally, the purpose of the Krebs cycle is to begin breaking down is to break down citrate. So you will hear this referred to as the citric acid cycle. Okay. So what happens when you start chopping off carbons? You get carbon dioxide. Okay. And through substrate level phosphorylation, where we take a substrate that has a phosphate on it and a high energy donor, donate that to ADP, we will get some ATP. So we get 1 ATP out of this process with 1 pyruvate. Okay, and we'll get 2 carbon dioxides out of it as well. And through these steps, we will eventually regenerate oxaloacetate. So we remove 2 carbons, right? What's left? 4 carbons. Perfect. Ready to join up with another acetyl-CoA. So far, we didn't get much out of this. We got some carbon dioxide, which is a waste product, and we get 1 ATP out of it. What else do we get out of this? And this isn't shown in the exact order that these come off in the Krebs cycle. I just was trying to show you what comes off in the Krebs cycle. The big thing we get out of the Krebs cycle is our electron carriers. These guys, the guys with backpacks. So now we've got, off of 1 turn of the Krebs cycle, 1 pyruvate, we get 3 NADH and 1 FADH2. And they're both carrying electrons and hydrogen in their backpacks. Okay? These are reduced electron carriers. So really the Krebs cycle is wonderful because it does produce a little bit of ATP, but it really produces these guys. These are the money. Okay, that's why we have the Krebs cycle. So why do we need those guys? We need those electron carriers because they have a job, big job in our last step here in glucose breakdown. And really at this point we've broken down glucose. Glucose, the carbons that are associated with glucose are not any part of what we're talking about right now. Okay, all we're interested in is what these electron carriers are going to do for us. So they're going to come over here, get a nice bright color like yellow. They're going to come over here to the inner membrane of the mitochondria. I think I forgot to mention the anatomy here of the mitochondria. Just as a quick review, that everything we've talked about so far has occurred here in the matrix of the mitochondria. Now we're moving to the inner membrane of the mitochondria. Remember, the mitochondria has 2 membranes, inner and outer. Okay, so this is the inner membrane here of the mitochondria. So we're going to move these electron carriers. They're going to go over here and drop off their electrons. Okay. Now remember, where are they dropping them off? The electron transport chain. That's what ETC stands for, electron transport chain. Okay. And the electron transport chain is a series of proteins, and I knew I drew them as these 3 membrane proteins here. In fact, there's more than 3. They all have names. They all have specific shapes. And that's not really vital for our understanding of this process to know what all of their shapes are. What I need you to know is that they have a job. Okay. When the electrons come in, the electron's energy is used to change the shape of the electron transport chain proteins. And when they change shape, because we know proteins are really good at that, when they change shape, they start doing their job. What is their job? Their job is to take all these hydrogens, also from the backpacks, right, and start pumping them. So they're a hydrogen pump. Okay? Where are they pumping them to? This space here is called the intermembrane space. To make it a little bigger, intermembrane space. Okay, so all the hydrogens are being pumped. Many of the hydrogens are being pumped through the electron transport chain proteins to the intermembrane space. The electrons are being passed. Remember, I said these guys are like a factory. They take those electrons, they pass them, and as they pass them, the electrons begin to lose energy. by the time they get to the end of the chain, it's a sad little electron here, okay, they need to be removed. Okay? If we don't remove those electrons, then we get this backup happening. If we don't get these guys out of here, new ones cannot come on board. So who takes those electrons out of there? This guy right here. This is why we need oxygen in this process. Oxygen is referred to as the final electron acceptor. It takes the electron from the electron transport chain, grabs some hydrogen as well, electron, hydrogen, oxygen, and makes water. Okay? So that water will become part of the cell's water, which will be in balance with the total body water. And the electron has been removed. And all is well here, so we can continue using the electron transport chain, which is great. So the question then becomes, why did we pump all these guys over here? So we got all these hydrogen ions filling up. So all these hydrogen ions have created, and pumping of these hydrogen ions has created a hydrogen ion concentration gradient. And that's great, because that means that if we open a door somewhere, these hydrogen ions, if it's appropriate, will flow through. And when they flow through, it acts like a current that drives something. Just like water drives a water wheel, these hydrogen ions are going to drive a protein. So let's take a look at that. So here they go. We're going to send them over here to this protein right there. I'm going to move our this way. Okay. So they're going to go to this protein. This protein is called ATP synthase. It is an, see the ACE on the end. Immediately your brain says that's an enzyme. Enzymes are proteins. So the hydrogen ions are going to flow through ATP synthase, and their current is going to cause ATP synthase to begin, part of it, to begin to spin, changing its conformation, and it does act like a turbine. And it is going, its job as an enzyme is to take ADP and phosphate, stick them together, and make ATP. Okay? So those hydrogens keep flowing through there, making lots and lots of ATP. So if we were to follow one glucose, we would find that, which would be 2-pyruvate, it would make at least about 30 or so, 30 plus ATP from this process. I forgot to use the very important word of what the flow of these hydrogen ions through ATP synthase, driving the production of ATP, is known as chemiosmosis. Okay? So those hydrogen ions flowing through, just like water flows through an aquaporin osmosis, these are hydrogen ions flowing through the ATP synthase, so it's chemiosmosis. And it produces a lot of ATP. Okay? So we call this process oxidative phosphorylation. Why is it oxidative? Well, as these guys are dropping off their electrons, they're in a reduced state, right? These guys are in a reduced state. So as they drop off their electrons and their hydrogens, they become oxidized. So we oxidize our electron carriers. We're also using oxygen to take those electrons out of there, and we phosphorylate a whole lot of ATP. So oxidative phosphorylation, referring to the oxidation of NADH and FADH2, and the high yield of ATP. So what do we do with all of that ATP? Well, we have an enzyme, wonderful enzyme here in the mitochondria, that will take the ATP. Some of the ATP might be used in the mitochondria, I'm sure, but most of it needs to leave the mitochondria, leave the powerhouse, right, to go, let's get another color, to go out and do jobs in the cell. It's not going to leave the cell. It's going to be used locally, recycled locally. It's going to be taken out of the cell, ATP out. Let's make that one of these guys. So ATP goes out, and ADP will be brought back in at the same time. So there's an ADP that needs to be recycled. It'll be brought back in for recycling. And there's an enzyme that does this. And it has a wonderful name because it describes exactly what it does. ATP, ADP, translocase. So you see the ACE on the end of it, you immediately think that's an enzyme. And what does it do? It translocates ATP and ADP, flips them. So we can recycle this guy and we can use this guy. Okay, so that's it for the big picture. If you have any questions, please feel free to contact me.